Hello guys, I'm Shifan. As we have only another 2 weeks for the exam, and this is the time students study really hard day in and day out. And in a way to help them out, I thought of making this video as a summary session on everything that we can expect for an essay in Unit 4 this year. What's till the end, as we will refresh everything that is important in Unit 4. Now, after thorough research and going through all the past papers the last 5 years, today I'll show you the 5 expected essay questions that we can possibly get on Unit 4. And I'll be going to each essay question and give you guys a full summarized revisit on each topic in that specific essay session. And another important thing, I have also made 2 videos on Unit 5 and Unit 2 expected essay questions. So if you haven't watched that yet, go through that too. The link for that will be given below. So let's get started. These are the 5 expected essay areas for this year's exam. First we have, these are all essays that we haven't got yet in A-level exams. First we have plant gas exchange, then water and solid movements. Here we'll take a look at diffusion, osmosis, bulk transport and everything else that is included there. Then loss of water in plants, which is transpiration. Then here we have 2 life cycles, pogonatum and salaginella. We might get one of these areas in our exam, but here let's go through both of them and let's revisit everything we studied in the life cycle. So let's get started with gas exchange. So what is plant gas exchange? Gas exchange is the movement of carbon dioxide and oxygen in and out of the plant during photosynthesis and respiration, right? Because plants carry out both photosynthesis and respiration. In both of these processes, gas exchange occurs. In photosynthesis, carbon dioxide is used up and oxygen is produced, and the opposite occurs in respiration. Mostly, gas exchange occurs through the stomata, but also sometimes gas exchange occurs through lenticels too. And also, cuticle is another structure that gas exchange occurs but in a very little amount. So we have 3 structures. First is the stomata, then lenticels, then cuticles. These are 3 structures that gas exchange occurs. And gas exchange occurs through diffusion of gases from high concentration to low concentration. So that is the basis of gas exchange. Now let us take a look at the gas exchange mechanism of stomata. We know gas exchange occurs because stomata is open, and stomata is open and closed by the guard cells. But how does this happen? How does guard cells open up and close during gas exchange? When water enters the guard cell, their turgor pressure increases, which means when water enters, water causes more pressure on the cell wall against the cell membrane. This increases the turgor pressure, causing the guard cells to expand. But guard cells are specialized in a way, they expand only from one side, which is the outer membrane. So the outer membrane expands because it is thin, but the inner membrane won't expand because it is thick. So the inner membrane bends while expanding. While the inner membrane bends, the stomata opens. This is the mechanism how guard cells open up the stomata. Again, when we lose water from the guard cells, the turgor pressure decreases. When the turgor pressure decreases, there is no force exerting. Because of this, the stomata closes. Now let us take a look at how the water enters the guard cell in the first place. So this is where the K plus influx hypothesis comes in. During daytime, hydrogen is present. Usually hydrogen is present inside the guard cell. But during daytime, this hydrogen is pumped out of the guard cell. When the hydrogen is pumped out of the guard cell, the guard cells become more negative. To balance this electrochemical gradient, potassium ions start to accumulate in the guard cell. When potassium ion starts to accumulate in the guard cell, the water potential inside the guard cells decrease. We know another thing is water travels from a high water potential to low water potential. Here in the guard cell, now we have lower water potential. So water starts moving inside the guard cells. Again, when the water moves inside the guard cells, the turgor pressure increases, opening up the stomata. Same happens uh, when the stomata wants to close. The potassium ions leaves the guard cell. Behind potassium ions, water also leaves the guard cell because the water potential increases. So when water leaves the guard cells, there is no pressure exerting on the guard cell. There is no turgor pressure, so the guard cells closes. This is the mechanism of K plus influx hypothesis. Now let's take a look at the role of abscisic acid in closing of stomata. So what is this abscisic acid? Abscisic acid is a plant hormone that reduces water loss when there is less water in the plant. So when there is less water in the plant has a lower water potential. This low water potential is detected by the roots and the leaves, which triggers the release of abscisic acid. This release of abscisic acid causes the pumping out of potassium ions from the guard cells. We know what happens when potassium ions are pumped out of the guard cell. The water in the guard cell leaves. This closes the guard cells. So, when there is less water in the plant, using abscisic acid, we can close the stomata. When we close the stomata, less water is released from the plant through transpiration. In this way, we can preserve the remaining water in the plant. So that's what abscisic acid does in closing of stomata. Now let's take a look at the factors affecting opening and closing of the stomata. Okay. There are four major factors that affects the opening and closing of stomata. First one is that stomata open at daytime and close at night. As I explained earlier, this occurs to the K plus influx hypothesis. On daytime, there is hydrogen ions. As I told, these hydrogen ions moves out of the guard cells and potassium ion moves inside the guard cell. Because of this potassium ions inside the guard cell, the water potential decreases. Because the water potential decreases, water moves inside the cell. When water moves inside the cell, the turgor increases. And because of this, the guard cells open. And the same occurs at night. At night, the potassium ions leaves the guard cells. Because of this, water leaves the guard cell. When water leaves the guard cell, there is no turgor pressure and guard cells closes. So that is the first one. And second factor that affects stomata is the concentration of carbon dioxide. Okay. We know on daytime, the stomata is open and photosynthesis takes place. Because of this photosynthesis, CO2 is always used up in the plant. This causes less CO2 in the stomatal cavity because the plants keep using up stomata for photosynthesis. This decrease in CO2 concentration causes the stomata to open more wider. So this happens during less CO2 and the opposite occurs at night time. We know at night time the stomata is closed, right? At night time photosynthesis doesn't take place. Because of this, CO2 is not used up. But at the same time, CO2 is generated during respiration because plants use up oxygen and release carbon dioxide. So CO2 starts building up around the stomatal cavity. This causes the stomata to close during night time. So that's the second factor. Then we have the internal clock of guard cells. As humans, we also have our own internal clock, right? We sleep at night, wake up in the morning. We have our own circadian rhythm. Same for guard cells. When the sun starts to rise, the guard cells open up automatically to carry out photosynthesis. And when the sun starts to set, the guard cells closes because it can't do photosynthesis anymore. So that is the internal clock of the guard cells. or the automatic closure and opening of the stomata. Finally, we have environmental stress. Changes in the environment can cause the stomata to close. So here we have drought, high temperature and the wind. We know when there is a drought, plants doesn't have much water in them. Because of this, water potential in the plant decreases. When water potential in the plant decreases, we know abscisic acid is released. Abscisic acid causes K plus to pump out of the guard cells, closing the stomata. So less water is lost through transpiration. Then we have again high temperature and wind. We know in both high temperature and wind, transpiration rate increases. When transpiration rate increases, more water is lost through the plant. To reduce this, the stomata is closed so that more water is preserved inside the plant. So this is everything about gas exchange. When it comes to an essay, this is everything that you have to write about. No more, no less, because every detail in gas exchange is explained here. Okay, now let us go through the second potential essay type question on Unit 4, which is methods of water and solid movements. We know when it comes to transportation of substances, there are two types of transport involved. We have active transport and we have passive transport. And when it comes to active transport in the resource book, they haven't given any separate paragraph on it. But they have mentioned in separate places where active transport is involved in plants transportation. So let us take a look at that also. And also let us go through passive transport. Okay. Active transport is involved in mainly three places. First, in the opening of the stomata using guard cells. We know to open the stomata, actually to open the guard cells, K plus is pumped inside the cell. Initially, even though K plus doesn't need any energy because initially it occurs through diffusion, but after some time, to create the turbo pressure, we need to pump more K plus into the cell. For this reason, we need active transport of K plus into the guard cells. For this, energy is used from photosynthesis which occurs in the chloroplast of the guard cell. Using that energy, active transport occurs which causes accumulation of K plus. Then the second place where active transport is involved is loading of sugar in phloem vessels. Okay, we know sugar is produced in the mesophyll by photosynthesis, and this sugar travels into the companion cells by facilitated diffusion. From companion cells to the sieve tube elements, where transportation takes place, it occurs through active transport because the sieve tube elements already have a high concentration of sugar. Because of that, sugar moves from low concentration to high concentration into the sieve tube elements. So this is another place where active transportation is involved. Then finally we have mineral uptake by roots. We know xylem sap in the roots have high concentration of minerals and the soil have a very low concentration of minerals. So to absorb minerals, roots use active transport using energy. So using active transport, minerals and ions are absorbed into the roots. So these are the main three places where active transport is involved in plants. So now let us take a look at where passive transport is involved. So when it comes to passive transport, we have diffusion, osmosis, imbibition, facilitated diffusion, and bulk transport. So let us take a look at each one of them. Diffusion is the movement of substances from high concentration to low concentration using energy or ATP. Even though movement of molecules, a single molecule is random, if you take a whole bunch of molecules, they move in a direction that is from high concentration to low concentration. And one of the major places where diffusion occurs is during gas exchange. During gas exchange, gases travel from high concentration to low concentration through the cell membrane. So this is one of the example for diffusion. Now let us go through osmosis. Osmosis is also another special type of diffusion. During osmosis, water travels from high concentration to low concentration. Uh, so here also it occurs through the concentration gradient. So one of the example for this, again, we have to go back to the guard cells. We learned in guard cells, after accumulation of K plus, water enters through osmosis from high water potential to low water potential. So that is one example of osmosis. Now let us take a look at imbibition. Imbibition is the adsorption of water by solid particles. This usually occurs in seeds. Most people confuse with absorption of water. So in imbibition, water is not absorbed into the substance or the cell. Water is adsorbed, which means water goes and bind to the surface of that substance and cause that substance to swell up. Even though water doesn't enter the cell fully, water travels to the molecules in that cell on the surface and causes it to swell up. And this is really helpful in certain situations, such as cause the seed to burst open and induce seed germination. That is one of the places where imbibition is involved. Now let us take a look at facilitated diffusion. Again, facilitated diffusion is same as diffusion, but molecules travel from high concentration to low concentration using transport proteins, such as carrier proteins or channel proteins. So one of the best example for facilitated diffusion is the movement of glucose from mesophyll cell to the companion cells. So we know, as I told earlier, sugar moves from the mesophyll cell into the phloem vessel. So between the mesophyll cell and the phloem vessel, we have the companion cells. So this sugar moves into the companion cells using facilitated diffusion, because the mesophyll cell have a high concentration of glucose than the companion cell. Then finally, we have bulk transport. Now, all this time, all the other passive transport, it occurs through short distance cell to cell. But bulk transport moves water and other substances to very large distances. For example, if you take a tree which is like 10 meters tall, it can't carry water through osmosis. So in this way, bulk transport is really essential for the transport of water. So in bulk transport, a pressure gradient is involved. If you take the xylem vessels, for example, where bulk transport occurs, transpiration pulls the water, right? Due to transpiration, water is pulled. This causes a negative pressure gradient in the xylem vessel. Because of this, bulk transport of water occurs through adhesion and cohesion of water molecules. And another example of bulk transport occurs in the phloem vessels. So when the phloem vessel is loaded with sugar, this causes the water from the xylem vessel to enter into the phloem vessels. Because of this, the turgor pressure increases, causing bulk transport of sugar molecules from the source to the sink. Sink is where the sugar molecules is needed for growth and nutrition. So these are two places where bulk transport is required, and this is all about the transportation of materials. So get prepared for that also. Yeah, I have everything that is required for an essay based on it. Now let's go to the next potential essay, which is based on transpiration. Okay guys, now the third important potential essay question for this year's exam is based on transpiration. Now when it comes to transpiration, we have three important parts: the process of transpiration, then we have stomatal transpiration, and finally we have the factors that affect transpiration. Now, factors that affect transpiration is not only important for the essay questions, but also for the MCQ questions and also for the structured essays. This is really important. Now, let us take a look at what transpiration is as a summary. Transpiration is the process where the plants lose water from the leaves as water vapor. This occurs through evaporation, and the major site where transpiration occurs is through the stomata. 95% of the water is lost through the stomata. So this was a question that was asked on 2024 biology paper. They asked about the rate of transpiration that occurs through the stomata or something related to that, I remember. But there was a question about it for sure. So always go through this part. Then transpiration also occurs through the lenticels, but a very little amount of water is lost through the lenticels. So how does transpiration occurs? So transpiration occurs from water traveling from high water potential to low water potential. We know where when there is high water vapor, there is a very high water potential, and water always moves from high water potential to low water potential. So because of that, transpiration takes place. Now let us go through stomatal transpiration. How does transpiration occurs? Where does it starts? It starts in the root. The root absorb water by osmosis. When when the roots absorb water, the water enters the xylem vessels, and through the xylem vessels, water travels upward in the plant. And how does this occur? This occurs through the transpirational pull of the water. So transpiration pull is the pull of water that occurs through evaporation, and water has certain qualities such as adhesion and cohesion, which helps in the transpirational pull. So when the water reaches when the water reaches the leaves, the water moves through the xylem vessels into the mesophyll cells. In the mesophyll cells, the water is evaporated, and also remember the mesophyll cell is moist and it has the highest concentration of water. So when it has a high concentration of water, there's a very high humidity. So in the mesophyll cell, there is 100% humidity, almost 100% humidity. So because of this, it causes a steep concentration gradient. When there's a steep concentration gradient of water vapor, it allows water to move outside through the stomata because the outside air has a very lower water potential and very lower humidity. Because of this, water moves out through the stomata. So that's it about stomatal transpiration. Is the whole process of how transpiration occurs. And now let's go through the factors affecting transpiration. So we have 6 factors that affects the rate of transpiration. So first we have light intensity. You know, light intensity increases during daytime because the sun rises on the daytime, which causes photosynthesis to take place. Do you know when photosynthesis is taking place on daytime, the stomata is open. So when the stomata is open, the rate of transpiration increases because more water is lost through the stomata. Then second, we have the temperature. When the temperature increases, evaporation rate increases. When the evaporation rate increases, transpirational pull of water upwards also increases. Because of this, more water vapor in the mesophyll cells, so more water is lost through the stomata. Third one, we have wind speed. So this is a bit more complicated than the other two. So when there is no wind, first let's take a look at that scenario. When there is no wind, uh, the humidity is very low in the outside air. So this is the surface of the leaf. Okay, and this is the mesophyll cell layer. Here we have the stomata. So okay, let me draw it in another color. So these are the stomata. Stomata are very close together. You know, there are a lot of them. So these stomata forms a shell outside the surface of the leaf. So let me draw that shell. So they form a very thin shell outside the leaf. Okay. So the humidity inside the leaf is almost 100%. So let me write it down. Okay. But sorry, and the humidity outside the air is 40%, which means there's very less water vapor in the outside air. So let me add that also. 40%. Now, because the inside of the leaf has a very high water potential, there is a very steep concentration gradient. Because of this, more water is lost to the plant. But Because of this layer, this surface layer formed by water vapor, we call it a shell formed by the stomata. Because of this shell has water vapor, this steep gradient decreases. Why? Because let's tell here we have 100%, here we have 40%. And in this surface, we have a 90%. Let's tell for example, let's tell we have 90% of water vapor. Because of this, the transpiration rate decreases because we have 90% here and we have 100% inside the leaf. So the diffusion gradient decreases. For this reason, when there is no wind, the transpiration rate decreases. But what happens if there is very high wind? When there is a very high wind, this surface layer is swept away. So there is no surface layer anymore. So when there's no surface layer, there's a very steep concentration gradient, 100% to 40%. Because of this, more water is lost through the plant. So during windy conditions, transpiration rate is high. When there is no wind, transpiration rate is low. Then we have humidity again. We know inside the plant the humidity is 100%, almost 100% inside the mesophyll cell layer, and outside the humidity is low. So transpiration is always high when the outside air humidity is really low, and it's the opposite when there is a very high humidity in the outside air. For example, now let's tell the humidity in here is 70%. Here 40% means very high transpiration, but if it's 70%, there is a very low transpiration rate. So as humidity increases, transpiration rate decreases. So that's about humidity. Then we have the concentration of CO2. Okay. Increasing of carbon dioxide concentration decreases transpiration. But how? We know when carbon dioxide concentration is high in the stomatal region, stomata is closed, right? Because carbon dioxide is only high when photosynthesis is not taking place. So stomata is closed. When the stomata is closed, water is not lost through the stomata. Because of this, the transpiration rate decreases. Finally, we have the availability of soil in water. So when there's less soil in the water, less osmosis of water takes place through the root. So root have less water and there is less transpiration pull in the xylem because there is no water. For this reason, there is less water vapor in the mesophyll cell, and if there is less water vapor in the mesophyll cell, the transpiration rate decreases. So that's about transpiration. Now let us take a look at the two life cycle. First one is the pogonatum, and then we have Salaginella. So let's go through that. Okay, guys, now let us go through the life cycle of pogonatum, which belongs to phylum Bryophyta. Now, when we take phylum Bryophyta, we know they are non-vascular plants. And in non-vascular plants, they don't have a extensive vascular systems, which means they don't have the xylem vessels or phloem vessels. Now, when it comes specifically to Pogonatum, they have alterations of generations, just like other plants' life cycle. They have an haploid generation of which is called the gametophyte generation, and a diploid called sporophyte generation. Now, specifically to Pogonatum, the gametophyte generation is dominant, which means they are photosynthetic, free living, and the sporophytes depend entirely on the gametophyte. So now let us go through the life cycle. Let's just summarize everything that we have done in life cycles. So this is the male gametophyte of Pogonatum, and this is the female gametophyte, and they are two separate plants. So this is why they call it dioecious or which means unisexual. They have two different plants. So this male gametophyte produces something called entheradia, which is the male sex organ, which produces the sperms. And the female gametophyte contains archegonia, which produces the egg. So this is the female gametophyte, and here on the top of the female gametophyte we have archegonia, which has the egg inside it. So now what happens is, at the right conditions, when water is present, these sperm cells, they travel through the water and reaches the egg cell. When they reach the egg cell is present inside the archegonium. The sperm cell fuses with the egg cell and forms a zygote. So when it forms a zygote, it starts the diploid generation, which is the sporophyte generation. So this zygote, so this is the zygote that is present in the archegonium, which is present in the female gametophyte. So this zygote develops into an embryo, which in turn develops into a young sporophyte. Also remember, the sporophyte has diploid number of chromosomes. So this young sporophyte grows on top of the female gametophyte. So here we can see this is the sporophyte and this is the gametophyte. The sporophyte is entirely dependent on the gametophyte for the nutrition and everything. Now the sporophyte, okay. So as I said, this is the female gametophyte and this is the sporophyte. This sporophyte has a capsule. So this we can see, this is the capsule which releases spores. These spores can develop into buds, and these buds develop into separate male and female gametophyte which have haploid number of cells. So again the life cycle repeats. So this is the overall summary of the life cycle of Pogonetum. Now let us take a look at the life cycle of Selaginella. Now unlike Pogonetum, Selaginella is a vascular plant which belongs to the phylum Lycophyta. And Selaginella has dominant sporophyte generation, which means they have diploid cells. And this is the sporophyte of Selaginella. And on the branches we have the strobilus. These strobilus has the megasporophylls and the microsporophylls. So in the megasporophyll it contains the megasporangium, and the microsporophylls it contains the microsporangium. So this microsporangium and megasporangium undergoes meiosis and produces the microspores and the megaspores. So when released, 4 large megaspores are released and numerous microspores are released into the external environment. So this microspore, which is enclosed in the microsporangium, develops into the male gametophyte, and the megaspore develops into the female gametophyte. So just like in the Pogonetum's life cycle, the megametophyte produces antheridium and the female gametophyte produces the archegonia. So during specific conditions, this antheridia produces sperms which travels to the egg which is present in the archegonia and undergoes fertilization and produces a zygote which is diploid, starting the sporophyte generation of Selaginella. So this embryo develops into a zygote and zygote develops into a young sporophyte plant. So the same cycle keeps repeating, and it is all for the Selaginella generation. Okay guys, that's all for today. So please go through all the essays that I taught today, and so it is about plant gas exchange, water solid movements, transpiration, and the two life cycles. So these are some of the expected areas, but always expect the unexpected. You never know what you'll get. So always go through the other life cycles and the other parts, and the xylem and phloem vessel part, and the phloem translocation. So all these are important, but give more importance to these areas. So that's it all for today. So see you guys. Thank you and bye.